Welcome, Catherine. Thank you for being here with me today. I hope you're doing great.
Yeah, thank you very much.
Thank you. So could you briefly introduce yourself and your role in Pancake?
For sure. So my name is Catherine Alix Panabière. I am a professor of cancerology at the University Medical Center of Montpellier in the south of France, and the director of the liquid biopsy and rare human circulating cells laboratory for many years now. I'm also visiting professor at the University Medical Center of Hamburg at UKE in Germany, as well as an active member and a work packet leader in the European Liquid Biopsy Society, which is named ELBS. And just to let you know that I have been working on circulating tumor cells, or CTCs, for 25 years. So my role in the European Consortium PANCAID is really to detect and characterize circulating tumor cells, as well as a large panel of circulating immune cells in different cohorts of well-defined patients in this consortium. So our goal with my EU collaborators is really to propose a well-defined blood signature defined as liquid biopsy to detect very early pancreatic cancer, the only way to save life in this very aggressive cancer.
Awesome. Thank you so much for being here. That's very impressive. So can you tell a little bit more exactly what is liquid biopsy and how do you plan to use this technique in Pancake project?
Yes. So the liquid biopsy concept was introduced for the first time for circulating tumor cells, or CTCs, as I mentioned before, in 2010 by Klaus Pantel and myself. However, to obtain a comprehensive liquid biopsy real-time view of cancer progression, we need to consider a broader definition of liquid biopsy that goes beyond CTCs and that includes now also first other tumor-derived circulating biomarkers, such as cell-free ctDNA for circulating tumor DNA, but also circulating cell-free RNA, non-coding or messenger RNA, extracellular vesicles like the exosomes or oncosomes, tumor-educated platelets, and proteins. But liquid biopsy includes as well tumor-induced circulating biomarkers, which are not released by the tumor, such as immune cells and circulating components of the immune system, such as cytokines, interleukins, but also circulating endothelial cells. So in addition, the liquid biopsy concept is not only related to blood, but has been expanded to other body fluids, such as cerebrospinal fluids, the urine, the bone marrow, the sputum, and even the saliva. And in 2023, I would like to mention that the liquid biopsy definition has been expanded again to the detection of the circulating microbiome in the blood, called liquid microbiopsy, which is the analysis of circulating cell-free microbial DNA in combination with a specific panel of proteins. So liquid biopsy is really complementary with the tissue biopsy, with data showing discordance between these two biopsies. Indeed, liquid biopsy can assess tumor heterogeneity and detect minor clones that can be missed in the primary tumor. In addition, we can detect clones or CTCs released from the primary tumor, but also from the secondary metastatic sites, which can be very informative to assess the clonal evolution regularly over time and during cancer progression. We know even that among these CTCs, a rare subset has stem cell properties and is able to initiate metastases, which is very important to better understand the metastatic process. As liquid biopsy is non-invasive, you can imagine that it can be performed before and then repeatedly and frequently during cancer treatment to propose a precision medicine to each patient. I mean, which drug we need to give to which patient and at the best moment. We talk about also real-time liquid biopsy, as CTCs and ctDNA have a very short half-life. It means that less than a few hours, than 2 hours, for example, meaning that when we detect these circulating biomarkers, we can be sure that they were just released by the tumor, which is very informative for the progressive status of the disease. And you talk about, of course, liquid biopsy is a revolutionary approach in cancer diagnostics.
This is really impressive. But what are the important areas of clinical applications of circulating tumor cells and circulating tumor DNA?
So clinically, liquid biopsies can be used first for early detection of cancer, using, for example, higher blood volumes. And of course, I believe that we need to rely on a panel of different but complementary circulating biomarkers will, without a doubt, increase the accuracy. However, today, screening is still very challenging. Second, tumor staging and monitoring of patients with localized cancer to discriminate patients at low and high risk of recurrence. Third, predicting metastatic progression in patients with localized and even advanced cancer. Four, identifying therapeutic targets to propose a personalized treatment to each patient. Five, monitoring on real time the efficacy of therapies and discriminating between early responders and non-responders to be able to switch very early and quickly to a more efficient drug. And finally, six, tracking tumor evolution with the identification of resistance mechanisms. So importantly, interventional clinical trials are crucial to demonstrate the clinical utility of CTCs and ctDNA, and even the other circulating markers, alone or in combination, and to introduce this innovative test in clinical practice. And this is really our goal, main goal in the European Liquid Biopsy Society, ELBS.
Thanks, Catherine. CTCs and ctDNA really have a wide range of potential applications. What about liquid biopsy and PanCAID?
We rely on the expertise of all the different partners in PanCAID, key opinion leaders in this field, to integrate numerous circulating biomarkers to identify the best panel for a minimally invasive blood test to predict and in turn prevent the development of pancreatic cancer. Our work package 2, in which my laboratory is involved, is fully dedicated to the detection of many different circulating analytes. For example, CTCs with different technologies, ctDNA with different approaches, metabolites, proteins, exosomes, and so on, to define the best ones to be detected in our blood signature with clinical relevance, to discriminate pancreatic cancer from healthy donors, but also from patients with a benign pancreatic disease. So we put a huge hope on liquid biopsy-based approach to detect very early a pancreatic cancer. It is crucial in pancreatic cancer where early detection is often challenging, but also will save life in future.
Thank you, Catherine, for this extensive but also concise overview of liquid biopsy and also your work. I think it was very understandable. Thank you for being here.
Thank you very much, Tamara. I'm very happy to be part of this EU consortium. Very fruitful discussion each time. And as I said, key opinion leaders in the field. So I believe that after this 5 years of work, we will find out something quite important for patients.